Good morning, Prof. IC. Assalamualaikum.
Assalamualaikum, Prof. Can you
Assalamualaikum,
hear me?
Prof. IC.
Salam. Nice to see you, Prof.
Yes, nice to meet you too. How are you?
Fine, Alhamdulillah.
Alhamdulillah, Alhamdulillah. We already sent the background, the visual background of our activity today, Prof. IC. I
Okay.
have sent
Okay,
you
let me
by
check.
WA. Yeah.
So, okay, yeah, please. Okay, you already gave me the backdrop. Okay, I will set up the backdrop first and then I will join again later. Okay, Prof?
Okay, okay, okay.
Okay,
Okay,
okay.
thank you.
Excuse me, before we start this event, can we wait for like about a couple of minutes before there are more participants to join? Thank you. For the participants, the students, can you all open your camera, please, and use the virtual background as been shared? Thank you. All right. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Oh, I'm sorry. Can all the participants please open their camera, please? All right, let me start over. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen, students and lecturers. May I introduce myself first? My name is Jordan Mesak. I'm a third year student biotechnology major at Diponegoro University, and on this fine day, I'll be your master of ceremony for the current event. which is our current event, is the visiting lecture for the World Class University program at Faculty of Science and Mathematics, Diponegoro University, with the theme Advancing Biotechnology Through Enzyme Technology and Protein Engineering, Public Health and Global Research Innovation. Now, before we start, it's good to start the day with a prayer based on our own religion and beliefs. Let us pray. All right, prayer is enough. All right, so before we start, let's have a photo first as documentation to memorize this event first. Can the participants please open their cameras so we can have our documentation session? All right. 1, 2, 3. Let's do it again with a different style this time. 1, 2. All right, that will be enough. All right, so without waiting any longer, let's start our visiting lecture with a welcoming speech from the head of the biotechnology major, Dr. Sri Pujianto. The time is and place is yours, sir.
Okay, thank you, Mas Jordan. Good morning, Prof. Dr. IC. Actually, I am Sri Pujianto, Head of Bioteknologi Study Program. Salam kenal, Prof. IC. Kebetulan beberapa waktu yang lalu kita juga pernah berkunjung ke University Pahang, ya. Waktu itu di pusat riset bioaromatik, bioaromatik ya. Eh, Baik, thank you very much for your time to share your knowledge for us in bioteknologi study program. This is very good opportunity for us to learn for you about the enzyme teknologi ya. Jadi Bapak Ibu dan juga para mahasiswa semua, ini adalah kesempatan yang sangat berharga bagi kita semua. Di awal semester baru kita sudah langsung mendapatkan sharing ilmu ya terkini tentang teknologi enzim. Kebetulan juga ada mata kuliah enzimologi ya. Kemudian ada teknologi enzim dan enzim industri. Saya kira ini kesempatan yang sangat berharga bagi kita untuk menimba pengetahuan dari Prof. IC yang memang ahli di bidang enzimologi. Selamat webinar, semoga nanti bisa banyak mengambil ilmu dari beliau. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Terima kasih Prof. IC.
Thank you so much for the and now for next is for an opening speech from the coordinator of this event, Professor Dr. Pancasakti Kusumaningrum. The time and place is yours.
Thank you, Jordan. Do you hear my voice clearly?
Loud and clear.
Okay. The Honorable Head of Biotechnology Study Program, Dr. Sri Pujianto, the Honorable Associate Professor, Dr. IC Nur Mazila Ramli from University Malaysia Pahang Al Sultan Abdullah as the guest speaker, and the Honorable all lecturers from Biotechnology Study Programs, and the others, the invitees, and all of the students who attend at this moment. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera untuk
Waalaikumsalam.
semuanya. Salam kebajikan, namo buddhaya. Good morning, ladies and gentlemen. Welcome to Diponegoro University, Prof. IZ. Thank you for taking the time to share knowledge and on this occasion as a part of world-class university program of Diponegoro University. Although we do it online, hopefully it doesn't burden our spirit to gain knowledge. And ladies and gentlemen, this visiting professor activity is held at the effort of Biotechnology Study Program to complete the performance target mandated by Diponegoro University. In particular, we hope that this activity can have an important impact, especially in increasing the knowledge of biotechnology for all students and all of the participants in general. We need to know about the advancing biotechnology through enzyme technology and protein engineering, public lecture, and global research invitation in order to provide us with information about this latest science and technology development. As we know that gene manipulation involves the use of biotechnological techniques to alter the genetic material of an organism, enabling the production of protein with desired characteristic. And recent advancements in protein and enzyme engineering have enabled the production of a diverse array of high-value compound and microbial system like enzyme, with the potential for industrial application. Hopefully, these visiting professor activities can be followed with the other broad activities in the form of cooperation on Tridharma Perguruan Tinggi from Diponegoro University and with University of Malaysia Pang Al Sultan Abdullah. That's all about the welcoming speech that we can confide. Thank you for your participation. Congratulations on joining this visiting professor activity. We hope that it will be useful for all of us. Please forgive us if there are any mistakes in our speech. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you for the opening speech, Professor. Now, for the rest of this event, it will be guided by our moderator, one of our lecturers here at Biotechnology, Bu Nurina Takta Afwi Maulina. The time and place.
Okay, thank you, Jordan. Is it my voice is clear?
Loud and clear.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Is it my pleasure to welcome you all to today's meeting, Visiting Professor Online. We are truly honored to have with us Associate Professor Dr. Aizi Nurmazila Ramli, the Dean of Faculty of Industrial Science and Technology, University Malaysia Pahang, Malaysia. Good morning, Prof. Aizi.
Morning.
Welcome to our meeting, Prof. Before we start the main session, please me to briefly introduce our distinguished invited speaker, Prof. Aizi. Okay, Associate Prof. Dr. Aizi Nurmazila Ramli received her Bachelor in Biology Industry, University Technology Malaysia in 2006, and also received her PhD in Bioprocess Engineering from University Technology Malaysia in 2012. And she currently serving as the Dean of the Faculty of Industrial Science and Technology from September 2023 and until now, and has previously held position as Deputy Director of the Research and Management Center, RMC, in University Malaysia Pahang, and also as a associate professor Malaysia Pahang. Her expertise, I'm so sorry. Her expertise cover enzyme biotechnology, molecular biology, computational biology, and also industrial microbiology. She has supervised, you can see on the screen, the numerous postgraduate and doctoral students receive multiple national and international awards, and also have so many international journal and publication. With such an impressive academic background and contribution, we are delighted to learn from her today. Okay, now without further ado, I would like to invite Prof. IZ to the main session of the main session. For Prof. IZ, the screen is yours.
Okay, thank you, Miss Nurina. Okay, so allow me to share my slide. Okay. Okay, can everybody see my slide?
Yes.
Yes.
Okay,
Prof.
let me. Okay, so thank you for your kind introduction. Thank you very much. It is a great honor for me to be here at Fakultas Sains and Matematika, FSM, University Diponegoro. So thanks to Prof. Sri Pujianto, Head of Study Program for Bioteknologi, and special thanks also for Prof. Dr. Hermin Pancha Sakti, the coordinator of the program. And then good morning to all the fellow lecturers here and all the beloved students. Thank you very much for inviting me. Saya sangat berbesar hati untuk bersama-sama dengan warga Universitas Diponegoro FSM specifically because of last week actually saya join juga satu seminar dengan Prof Sugiharto di fakulti yang berbeza. So again, it is my pleasure to be here to share some information, some knowledge. I'm not so good actually, tapi saya harap my sharing session today will be benefit to all of you for today. So as moderator mentioned, yes, I'm IZ. You can call me Dr. IZ or IZ. So I'm from Faculty of Industrial Sciences and Technology, University Malaysia Pahang, Sultan Abdullah. So now, currently, I'm the dean of the faculty. So yeah, let's continue to the next slide. So because of, I'm aware that when I received the invitation letter, so it is mentioned that for the today's audience is actually from the lecturer and also from the student. So I hope that the lecturers can bear with me because I would like to explain a little bit about the theory so that the student can catch up with me. So I would like to recall the definition of an enzyme. So again, lecturers, ibu-ibu, bapak-bapak, please bear with me. This is a short warm-up for the students. So I would like to explain about the enzyme. So what is the enzyme? So enzyme is actually the biological catalyst. So it is a protein, but sometimes it can be the RNA that speed up the biochemical process. How? By lowering the activation energy. So it is the minimum energy required for the catalyst to convert the substrate into the product. So enzyme is a special, it is only come from the living cell. Okey, kita tak jumpa enzim ni dekat benda yang tidak hidup. Kita tak jumpa dekat kertas, kita tak jumpa dekat meja. Enzyme can only be found from the living cell. For that reason, we call it as biocatalyst. And then it will lowering the activation energy, which is the minimum energy for the catalysis to occur. And then it will speed up the conversion of the substrate into the products. Okay. So it is a reusable helper. Okay, what is the meaning of reusable? Means that enzyme can be used again and again. Okay, if there is the substrate in the reaction mixture, so enzyme will work until all the substrate converted into the product. So this is the special thing about enzyme. Okay, however, enzyme needs special requirement. For example, pH and temperature. Sama juga dengan kita allow me to mix bahasa and also English, okay? Is it okay for you? Can I
Okay,
hear from
okay.
student? Okay, nice. Okay, so
Okay, Prof.
okay, so means that same with us, enzyme also needs special requirement. Okay, they need special condition including the temperature and pH, which I will explain further in the next slide. So as you can see here, so this is how the enzymes work. So you can see the substrate here, and then it is like a mouth in the enzyme, which we call it as the active site. And the shape of the active site is similar to the shape of the enzyme. So the substrate will accommodate the active site, and then the catalysis will happen. So the enzyme will weaken the bond of the substrate so that the substrate will be converted into the other form that we call it as the product. So the blue one, okay, as you can see here, is the product, okay, the reaction product. So we call it as the recognition and binding. So it is like the lock and key, okay, where the shape is we can recognize the shape. I mean the enzyme can recognize the shape, and then it will form the enzyme substrate complex. So the middle one is the form of the enzyme substrate complex, or we call it as the ES form, before it will release the product into the environment. So why the product release into the environment? Because of the shape of the product cannot fit longer into the active site of the enzyme. For that reason, it will release into the environment, and then enzyme will do its work again by try to find the other substrate and then convert the substrate into the product until all the substrate finish. Okay, this is how the enzymes work in the reaction mixture. Okay. Okay, I hope so far is okay. So before I move into the next slide. So this is the explanation about the application of enzymes. Okay. So one toolkit, many markets. So this is what I want to show here, how a single class of molecules that we call it as enzyme powers our everyday industries, including the food processing, pharmaceutical, biofuel, and also the cleaning products. So as you can see here, for the food processing, I put the example of lactase, where this lactase can cut or convert lactose into the simple sugar. So it is very important actually for the lactose intolerance people, because lactose intolerance people can have lack of lactase in their body. So if they consume food with the lactose, so they will face some tidak selesa in their body, in their stomach. So for that reason, the food, okay, for the safe conditions, so it is good to for the enzyme lactase, okay, to convert to have the lactose intolerance product. And then it is for us, for the normal people, lactase in our body, when we eat the lactose food, so this lactase is responsible to convert the lactose into the simple sugar, so that we can get benefit from the food products. Next, I move into the pharmaceutical, where here I put the enzyme amylase. So this amylase enzyme can help for the formulation and digestion aid, and also diagnostic. So it is a reliability for the specific reaction with minimal byproduct that can simplify the purification. So this is the example of the enzyme product for the pharmaceutical, which is amylase. Now I move to the cleaning product, where I put here protease. So actually protease is also incorporated into detergent product, because of protease is specific for the protein. means that there are a lot of stain that contain the protein. So by including protease into detergent product, so this detergent can do the cleaning process efficiently at low temperature condition. Okay, so a lot of enzyme actually have been put into the detergent product. So one of them is protease, but other than that, you can find amylase, lipase, that a part of bioactive compound in the detergent products. Okay. Now I move into the biofuels. So as you can see here, we can convert the biomass into the biofuels. So the example of the enzyme is the cellulase. Okay, cellulase can convert the cellulose into the simple sugar and then can be used for the biofuels development. Okay, still in the theory slide, okay, because again, okay, because I'm aware that there are students here, so I would like to give you some theory. Okay, so I would like to explain a little bit about the factors that can affect the enzyme's activity. So as you can see here, sama dengan kita perlukan special equipment special necessary special things. Okay, for example, kita perlukan suhu yang sejuk, suka duduk dalam aircon, buat kita rasa selesa. So same goes to enzymes. So enzymes require optimum temperature for them to do the catalysis. Lower than and higher than its optimum temperature, it will denature or unfold. Okay, so this is the special requirement of the enzyme. So for that reason, if you want to use the enzyme for the industry, the first thing is to know their optimum temperature so that you can control the enzyme. Let's say you want to stop the reaction. Contohnya, jika kita tidak mahu the reaction happen, so we can lower the temperature or increase the temperature. So for that reason, the reaction will stop. Okay, so because the condition is not optimum for the enzyme. Okay, same goes to pH. Enzyme also require optimum pH, where at certain pH, the catalysis happen optimally. Lower and higher than that, okay, it will interfere the enzyme structure and then it will lead the product formation stop. Okay, next, the other factor is the enzyme concentration. So basically, if you increase the enzyme concentration, it will also increase the reaction rate. So this is the theory of the enzyme catalysis. Okay, so the increased enzyme concentration, okay, this enzyme will find the substrate and will convert the substrate into the product. And then lastly is the substrate concentration, where the substrate concentration also play a significant role for the catalysis to happen. So if there are a lot of substrate, at some point the enzyme will become saturated and the product formation will be stagnant. Okay, so this is the basic things that students need to know, okay, before you want to proceed into protein engineering. Because if you want to engineer the protein, you need to know what is the things that you can manipulate, what is the thing that you want to change, okay, or you want to mutate. So this is the things that you need to understand before you go further into enzyme engineering or protein engineering. Okay? Okay, let me continue. I hope you're still here with me. Okay, so I would like to explain a little bit. Okay, this is the example about the digestive enzyme. Okay, which is the enzyme that will do or that will degrade the food that we eat. Okay, for example, here I will repeat the theory where the enzyme have the specific active site that will recognize the specific substrate, and then will form the enzyme substrate complex. And after that, this enzyme substrate complex, the catalysis will happen at this form, and then the product will release into the environment once the product will not longer fit into the active site of the enzyme. And then what happens next is the enzyme will find another substrate for the conversion of product to happen. Okay? So as you can see here, this is the basic thing happen in our body actually. If we eat the carbohydrate, for example, pagi tadi ada makan nasi, ada makan tempe, okay. So what happened actually, this food product will be converted by the carbohydrates enzyme into the simple sugar. So the carbohydrates enzyme including amylase, maltase, lactase, and sucrase. So this is the reason why, okay, once we eat the food in our in our mouth, there is in our saliva actually contain the amylase. So this amylase will degrade the starch into glucose. So there is the reason every time bila kita makan, kita rasa manisnya makanan itu. Because of the glucose itself is the sugar. Okay, so this is the basic theory. And then if we eat the protein also, the other enzyme will react with the protein, which is known as protease or proteolytic enzymes. This protease can be the pepsin, trypsin, and peptidase will also convert the protein into the simple sugar so that our body will absorb the nutrition, okay, for the metabolism and for our growth, for our nutrient uptake, and so on. And then lastly, fats. For example, ada minyak dalam makanan kita atau ada lemak. So this fat will be converted by the lipase into the fatty acid, simpler fatty acid. Okay, this is the basic theory. Okay, enough for the basic theory of the enzymes. I hope that student can gain the information from here, because after this, I will explain to you about the study case that happened in UMPSA. Okay, next, I would like to explain the next topics, which is the protein engineering. Okay, so the protein engineering is actually the most common things that scientists talks nowadays. Okay, so for the basics, it is to faster the reaction. Why the protein engineering is important? Okay, so the protein engineering is important basically to faster the reaction. So some of the biological catalysts can be done quickly. And then other than that, we also can manipulate the enzymes, or we can change the enzyme so that it can work in gentle condition. For example, we can run the reaction at lower temperature, lower pH, and by using fewer chemicals. What happened actually in the industry, they usually use harsh condition. For example, a very acidic condition, very alkaline condition, and and also they require the hot temperature, okay, which is the heating. So all of these things actually will cause the capital cost to increase. So for that reason, industri tak mahu capital cost increase. Dia akan melibatkan cost, dia punya operation akan meningkat. So basically this is not the best thing to happen in the industry. So by using the biocatalyst, which is the enzyme, so it will allow the reaction to happen at the lower temperature. So tak perlukan heating process at the mild pH condition, and they will also allow the fewer chemicals to be used. So this is actually the purpose of protein engineering. We can change the native enzyme, okay, so that it will work at what we want. Okay, kita boleh ubah that enzyme so that kita, for example, the reaction need to be happen at 15 degrees Celsius. So we change the enzyme, we mutate the enzyme, so enzyme will perform the catalysis at that kind of temperature. Okay? Okay, so let's move to the next one. So other than that, we also can create the enzyme which is more robust. Means that it is more durable, okay? It can work at harsh condition, and then it can be reusable also. Okay, and then other than that, the importance is the targeted action where it can improve selectivity, specificity, so less byproduct will be produced. This is the other things about the protein engineering, where we can enhance the condition of the enzyme so that the enzyme will work very specific, so that it will reduce the formation of the byproducts. So actually in the industry, the byproduct formation is also the things that we need to reduce, Because of other than the product, our target product, industry also need to handle the waste. Okay, selalunya kita tak boleh release the waste to the environment because the ministry won't allow it to happen. Okay, so for that reason, all the byproduct from the industry need to be treated. So this treatment also require the cost. Okay, so kebanyakan industri kalau dekat Malaysia, sekarang saya dekat Pahang, so ada banyak multinational punya company dekat Gebeng. Nanti kita cakap pasal student mobility, mungkin student boleh datang sini, kita bawa ke industri punya tempat dekat Gebeng. So that side memang ada banyak kilang-kilang. So when I try to discuss with them about their problem, the main issue kadang-kadang adalah the product itself. Satu benda, tapi satu lagi adalah they produce also a lot of byproduct or waste. So this waste, they need to treat the waste before they can release to environment, and this is also cost them a lot of cost. Okay, so this is the things we as a biotechnologist need to think, which is to cater the problem of the industry. Okay, and then new capabilities. Okay, so that the protein can have the new reaction, can work at new condition and so on. So this is also the great thing about protein engineering. And then lastly, the lower cost. This is the thing that I explained just now, which is to reduce the cost of the industry player, I mean. Okay, let's move to the next slide. So this one, I try to show the students about the concept of the protein engineering, where as direct evolution, and it's okay because it's known as direct evolution and computational design, where this will lead into the production of mutants, of new mutants that will benefit a lot of application. Okay, for example, pharmaceutical, food and beverage, and many more. Okay, let's move into the next slide. As you can see here, okay, this is the rational design where it requires a lot of in silico study. In silico means that you will use the prediction from the computer, okay, before you proceed into the mutagenesis production. Okay, so students, I hope you can see at my left hand, okay, kat sebelah kiri saya. So it is about the rational design where it's required the aid of the computer analysis for the prediction before we enter the lab to do the mutation. So for the mutation, we call it as site-directed mutagenesis, okay, which is, it is the rational design. From the computer analysis, we know how to improve the protein or the enzyme, and then it is based on the rational design. Okay, after that, we do the transformation, which is step number 3, before we proceed into protein expression and protein purification, which is the downstream process. And then we will get the desired mutant, so usually single one, okay, before we proceed into biochemical testing, and if the mutant is successful, we can use it for the industrial usage. Now, let's move into my right hand. Okay, sebelah kanan saya, the other approach of the protein engineering, we call it as directed evolution. So the good thing about directed evolution is that it is not require any structural analysis. Okay, you tak perlukan komputer, tak ada komputer pun tak apa, terus masuk laboratory ataupun masuk makmal. And then in the laboratory, you will use the random mutagenesis, means that you won't know the location of the mutation. Compared to the rational design, you tahu kat mana yang you nak do the mutation. For the random mutagenesis, the mutation happen randomly. After that, you will proceed with the transformation and protein expression, and the good thing is that you don't require any purification process. You don't need to purify your protein, but you require the screening and selection process, which is usually very tedious. Okay, so for this directed mutagenesis, you will get a lot of mutants usually. Okay, you will get the selected mutants. I mean, you will get your desired mutant will be more than one, and then you will proceed with the biochemical testing, and then you can use your mutant for the industrial application. Okay, so based on my experience, I already performed the rational design. So rational design, the struggle is actually during the computer analysis. It will take a lot of time for you to do the in silico study, okay, because we want to see, we want to predict what happened using the computer or dry lab before we enter to the wet lab. And after that, I perform the site-directed mutagenesis and I improve my protein before I use the protein for the downstream process and also for the industry. Okay, this is based on my experience. Okay, any questions so far? You can ask me the question if you want to ask. Boleh je selingan. Saya harap boleh faham. Okay. Now we go to the next slide, okay, which is from theory to lab validation. Okay, so this is where I would like to explain about my findings, okay, at UMPSA. So at UMPSA, we are committed to advancing biotechnology through enzyme technology and protein engineering. So we conduct studies to characterize the enzyme from locally isolated microbes and also from agriculture byproducts, evaluate the performance and enhance their properties. So this is, so for the next slide, I would like to show you some of the real case study based on my research experience so that you will know, okay, what happened, what you can do, okay, for the field of the enzymology and also for the field of the protein engineering. Okay, let's move to the next slide. Sorry. So yeah, actually, the moderator already explained a little bit about me actually. So yeah, my field is enzyme technology or enzymology. So I'm involved with enzymes since 2006 to present. So I can say, I can categorize myself as enzyme technology researcher. So early work, I work with the cold adapted enzyme, which is this enzyme is isolated from Antarctic, okay, and it is funded by Ministry of Science and Technology Innovation, Malaysia. Okay, so yeah, not me, my team go to the Antarctic to do the isolation of the yeast that we call it as Glycosoma antarctica. So from there, we studied a lot of cold adapted enzymes and how this enzyme can be used for the industry in Malaysia. And then for my core skills is enzyme technology. I'm also doing a lot of the computational biology, for example, structural prediction, docking, and mutational scanning. Also include the molecular dynamics. And then for the protein engineering, as I mentioned from the previous slide, I'm doing the rational design. And then I also involved with the stabilization and immobilization using the cross-link enzyme aggregates. And I also involved with the design of experiment-driven process and product optimization, including the formulation and reusability. And then for the natural resource focus, I work with bromelain from Malaysian pineapple cultivars. I also work with Acolera melecensis or agarwood, which is involved the enzyme resin pathways. And then I also work with edible and medicinal mushroom, for example, cordyceps, Linosus rhinoceritis ataupun cendawan susu harimau, and Pleurotus species, which is cendawan tiram in Malaysia. And for enzyme-assisted extraction, metabolite mapping, and also fermentation scale-up, I also work with the tropical biomass valorization, for example, from the pineapple byproduct to the functional ingredients. I try to find the alternative of the prebiotic actually, okay, for the agriculture purpose. And I have IP, for example, IP means intellectual property. For example, for meat tenderizer, natural liquid cleanser, which is the cleanser actually formulated from the bromelain, okay, which is from the leaf of the pineapple. And then I also developed the product which call it as pineapple mushroom product, okay. Okay, let's move to the next slide. So other related study also in enzymology, okay, which is as I mentioned agarwood or the karas tree, or kat sini kita panggil pokok gaharu. Okay, in Malaysia we call it as gaharu, which is comes from Acarnaria species. I believe that a lot of Acarnaria species also from Indonesia. And then after that, I also did some optimization of edible bird nest cleaning process using immobilized enzyme. And then I also work with some enzyme isolation from Malaysia local environment. And then I also studied some phytoenzymes from local fruit, for example, durian and also jackfruit. So this is the things happened to me for the last 10 years. Okay. So, as I mentioned, I would like to share a little bit about my research study because of I believe that a student cannot rely only on theory. They want to see what happened, what is the application. So after that, maybe if student want to work with government or they want to become the entrepreneur, this is the thing. Use your theory to benefit others. So this is the things that you can consider. So what happened actually at UMPSA, this is the things that we develop, where we try to use the theory of enzyme technology into the commercialization. So the first thing that I would like to share today is the enzyme usage for edible bird nest, or we call it as edible EBN, bioprocessing. So for your information, okay, Swisslet, the edible bird nest comes from Swisslet, okay. So it is the high value delicacy and also traditional health food. Okay, it is mainly consumed in China and also Southeast Asia. So basically, Malaysia and Indonesia are the top global producers and exporters of edible bird nest or Swisslet. So edible bird nests are rich in glycoprotein and also epidermal growth factor (EGF), and also salicylic acid and neuraminic acid. So this is the things, the profit of eating edible bird nest. Okay, so some challenges in the industry that labor intensive and manual cleaning process. What happened in the industry? So usually they will try to clean edible bird nest by picking one by one the feather of the bird. Okay, and also sometimes the poops and also the soil that trap inside the edible bird nest. So this is really time consuming and require a lot of labor. Okay, so sometimes also, okay, to speed up the process of cleaning, they will use the chemical, for example, the bleaching in some operation. This is not good because for the human health. And then there are concerns over residue, hygiene, and halal compliance, and lack of standardized, scalable, and eco-friendly technologies. So for that reason, to maintain international quality standard and meet rising demand, innovation for the sustainable processing solution are needed. So what happened at UMPSA? So we developed clear keratinase from Swisslet feather enzymes. So we improved the thermal and pH stability of the enzyme. And we found that these cross-linking enzyme aggregates, okay, can be reusable for multiple cleaning process. So based on my study, we can use the same solution, okay, of the keratinase up to 5 times. So this is the good things about the immobilization technology. Selalunya kalau kita pakai solution yang ada enzim sahaja, so selalunya kita akan ada kesukaran or limitation to separate between the product, which is the edible bird nest, and also the enzyme. Okay, because they are contained in the same solution and they are usually soluble. So for that reason, it will increase the cost because enzyme itself is actually very expensive actually. So by using the immobilization technology, which is the technology that I use is the cross-link enzyme aggregate, where I link the enzyme together using the linker, which is glutaraldehyde. So this glutaraldehyde is like a rope that will tie the enzyme together. Okay, dia akan ikat satu enzim ke satu enzim lain, dia akan jadi satu gumpalan, and this enzyme will work to clean the edible bird nest, but at the end of the process, we can separate the enzyme and the edible bird nest for the next cleaning cycle. Okay, this is the good things as I mentioned about the immobilization technology. At the same time, this immobilized enzyme also is very stable compared to the free enzyme. So I put here in this slide the advantages of clear keratinase. Keratinase is basically the enzyme that will degrade the keratin. Keratin is the main component of the bird feather. Okay, so it is cost effective as I can reuse the same solution, maintain the high activity across several users, reduce the enzyme degradation as the enzyme is more stable compared to the free enzyme, and it is suitable for semi-automated industry for the scale application. Okay, let's move to the next slide. Okay, this is actually the infographic about the immobilized keratinase for efficient and recyclable cleaning of edible bird nest. As I mentioned to you, I extract the enzyme, I immobilize the enzyme, and I use the enzyme for the cleaning purpose. And this enzyme, some basic properties of this clear enzyme is that it is eco-friendly, high stability, and can reduce the cost. Okay, it is also safe compared to the chemicals. Okay, so any questions so far? For me, saya laju sangat ke? I hope you are okay with my pace. Boleh? Okay, let's move to the next study case that happened in UMPSA. Okay, which is coal power enzymes that love the chill. So I would like to talk about the cold adapted enzyme as I just mentioned in the previous slide. I mentioned about my first study about enzyme is actually the enzyme that have been isolated from the Antarctica. Not Antarctic actually, Antarctica, which is the yeast, okay, which is isolated from the cold environment. And we categorize this kind of microorganism that can thrive at cold environment. We call it as psychrophiles. And then these psychrophiles, in order for them to stand the lower temperature, they will have the special properties, which is their enzyme can adapt or can stand at lower temperature. This enzyme, we call it as cold adapted enzymes. Faham ya? So dekat cold environment ada banyak microorganism. So this microorganism kita panggil dia sebagai psychrophilic ataupun psychrophiles. These psychrophiles, macam mana mereka boleh stand untuk thrive ataupun untuk hidup dekat cold environment is by having the special set of enzyme. And this special set of enzyme, we call it as cold adapted enzymes. So this cold adapted enzyme, when we compare to the mesophilic enzyme or the enzyme that can thrive at ambient temperature, they have several properties or several unique properties. For example, as you can see in this figure, okay, so they have the reduced hydrophobic and electrostatic interaction that make them very flexible. This enzyme dia sangat fleksibel. Kalau enzim yang ada daripada mikroorganism yang hidup dekat thermophilic punya environment ataupun hot environment, dia enzim is usually rigid. Dia punya enzim tu tak bergerak, kaku. But for cold adapted enzyme, dia lebih fleksibel. Okay, because of for them to stand at lower temperature environment. Other than that, they also found to have reduced hydrogen bond, salt, and also disulfide bridge. So this is the things that we study from our computational study. We found that this is the unique properties about the cold adapted enzymes. Other than that, they also have the greater surface hydrophobicity, okay, wide substrate pocket size, okay, dia punya active site itu adalah lagi besar. And then they also contain lower arginine and lysine ratio, but increased glycine residue. So this is the other things. Arginine and lysine is actually a big or more complex amino acid. So for the cold adapted enzyme, usually they will have the simple amino acid like glycine and also alanine. Okay, they will also have a lower proline. Proline is also a very complex amino acid which have the proline or aromatic residue. So this proline usually found more in thermophilic enzymes. Okay, because of proline will increase the rigidity of the enzyme. Okay, it will restrict the movement of the enzyme. So for the cold adapted enzyme, they don't need the proline. So they will, if you study the cold adapted enzyme, they will have the lower proline content. Okay. So as I mentioned to you, our sample is known as Glycosema. Oh, sorry, this is not Glycosema antarctica. This is Glycosema martini. So it is yeast actually, okay, which is categorized from the fungi. Okay, which is the unicellular cell, with the unicellular cell. Okay, let's move to the next slide. Okay, so what happened, okay, for the Antarctic yeast, Glycosema antarctica, strain PI12, it is the cold adaptation signature. Okay, what we study actually, the cold adaptation signature in Glycosema antarctica PI12, which is including the chitinase enzyme, beta-manganese enzyme, and also alpha amylase enzyme. So what we did previously, we do the cloning and expression, and we also study the biochemical characterization at low temperature. We also analyze the kinetic and stability profile of this cold adapted enzyme. And lastly, we try to sequence and do the structural analysis. So what we found is actually, okay, the cold adaptant signature across the enzyme. For example, a higher loop flexibility, fewer stabilizing salt bridge or hydrophobic content, conserve catalytic cost, and they perform the catalytic activity at lower temperature. For my case, the chitinase enzyme and the amylase enzyme work best at 15 degrees Celsius, okay, with the usual trade-off in thermal stability. They are not very stable in the high temperature. Okay, let's move to the next slide. Okay, I hope you're okay with my presentation pace, but you can inform me kalau saya laju sangat. Okay, so this is based on my findings. Okay, so I would like to show you some of the structural analysis. For this one, I use the computational modeling to predict the structure of the chitinase enzyme from the psychophilic Glycosema antarctica, and I try to analyze its structural properties and function. As you can see in my left hand, okay, yang structure warna biru dengan kuning, I'm referring to this structure which is known as chitinase. A is actually the enzyme, the chitinase enzyme that we found at the mesophilic temperature, at ambient temperature. And B is my targeted enzyme, which is isolated from the psychophilic Glycosema antarctica. we found that this chitinase from Glycosamine antarctica, which is categorized as cold-adapted enzyme, is very flexible actually. They have the shallow active site, and they have a wide range of the active site specificity. I mean the substrate specificity. So this make them able, okay, to stand the lower temperature compared to structure A, which is the mesophilic structure. So, however, this enzyme, when we expose them at ambient temperature up to 25 degrees Celsius, this enzyme will denature. Okay, they only work best at lower temperature, which is 15 degrees Celsius. However, beyond than that, this enzyme cannot perform catalysis. So this is the unique thing about the cold adapted enzymes. Okay. So let's move to my right hand, which is the structure, yellow and red structure. So this is the beta manganese, also isolated from the same organism, which is Glycosamine antarctica. As you can see here, the A, which is the beta manganese isolated from the ambient temperature or mesophilic temperature, as you can see, it is more compact. Lagi besar kan? Okay. So, and it is more rigid. Okay. However, the B, okay, B structure, as you can see, contain a lot of loops. So this loop region will give the flexibility properties to the enzyme, so the enzyme can withstand the lower temperature. Okay, so this is the basic difference or the common difference that we can explain, we can differentiate between the high temperature enzyme and low temperature enzyme. Okay, next we move to the lower figure, okay, which is the amylase enzyme, which is isolated from the same microorganism. As you can see also, we found the novel properties of this enzyme where the pocket, the active site of the enzyme contain several unique amino acids that differentiate them between the mesophilic enzymes. Okay, so this is basically the study that we done in our lab for the cold adapted enzymes. Okay, let's move to the next slide. After this, I will open to Q&A. You can ask me, okay, whatever you want to ask or you want to clarify about my research findings. Okay, is it okay? So far, okay, students? Are you okay with my explanation? Can you understand me? Boleh
Okay,
ke?
Prof.
Okay, nice. Jordan, do you understand me? Can you catch up with my
Yes,
lecture?
Prof.
Yes.
Lepas ni I tanya Jordan Pop quiz. Okay, next. We move to the next slide. Okay, now we move to my other case study, which is the catalytic to commercial modeling bromelain to use case. So I also studied about the bromelain. As I mentioned to you, I study from the variant cultivars of pineapple in Malaysia, and I also try to extract the enzyme, which is the bromelain, which is very specific. Bromelain can only be found in pineapple. So for that reason, I extract the bromelain, which is mostly found in their byproducts for the commercialization, where I try to develop the product or the innovation from this bromelain. Okay, so I would like to share a little bit about the global market value of bromelain. As you can see here, the price is increasing year by year, and most of the bromelain can be found in the pharmaceutical products. And then bear in mind, there are two types of bromelain, which is stem bromelain and fruit bromelain. And these two types of bromelain will give you different benefit. Okay, stem bromelain, which is the bromelain that is isolated from the stem part of the pineapple. And the fruit bromelain, we can get this kind of bromelain only from the fruit part of the pineapple. And usually the stem bromelain will give you a lot of properties for the therapeutic punya function. Okay. And then this is what happened to my lab analysis of bromelain from different pineapple cultivars. As you can see here, I try to study, I try to use 5 cultivars, which is commonly found in the market in Malaysia, which is Josephine, Sarawak, MD2, Morris, and pineapples. But I don't know whether this cultivars is similar to Indonesian. So, boleh tak saya tahu dekat Indonesian selalunya dia punya cultivars atau variants-nya nama apa? Mungkin Prof, ada nama lain ke Prof? For,
We call it only pineapple, but there is a lot of varieties in Indonesia. But
I,
for the local name, we do not know
I
actually,
see.
because I do not work with this pineapple cultivars. Thank you.
Prof suka makan pineapple?
Yes, yes.
Okay.
We have a kind of cultivars that's very, very
Hmm.
the taste is it's very nice, but it is very small. We
Okay.
call it nanas madu.
Oh, nanas madu. Yes, we also have the nanas madu.
Oh,
But
the
in
same,
Malaysia,
yes?
it is under Sarawak. Yeah. Actually,
Oh,
if we
okay,
want to
okay.
differentiate the pineapple, okay, Prof, and also the audience, actually,
Okay.
kita boleh lihat dari dia punya crown. So, crown is different actually. So, this difference of crown is because of different of the pineapple cultivars. So, in Malaysia, we have a lot of cultivars. And the most famous one, yang ni sangat manis. Okay, nanti kalau Prof Hemin datang Malaysia nanti, saya bawa makan MD2. Dia sangat manis.
Insya Allah. Nanti barangkali ada masa mau ikut. Jordan mungkin mau ikut.
Yeah, usually when we
Terima kasih, Prof.
when we eat a pineapple, usually we will have the sensation yang kita rasa it's not very nice at our tongue. It is because actually of the content of the bromelain actually. Because of the bromelain is actually categorized as proteolytic enzymes that will degrade the protein. So what happen bila kita makan nanas, kita akan rasa tidak sedap dekat lidah because of the action of the enzymes. But for the MD2, it is nice. It's not really sensitive to our tongue. And it is also very sweet. So this is actually the most common pineapple variants or cultivars in Malaysia. So I try to do some study in order to analyze the content of the bromelain from these different cultivars. And interestingly, I found that MD2 contain a lot of bromelain. So it is good if we consume MD2. And then after that, I try to study the structural analysis of this bromelain, which is A is the stem bromelain and B is the fruit bromelain. So from here, I can see the difference of action between stem and fruit bromelain. Okay, next. So as again, I would like to explain a little bit about the application of bromelain in the pharmaceutical field. So why we want to extract this bromelain? Because of their potential for the pharmaceutical field, which is bromelain uniquely only found in pineapples. So it's a valuable and most extensively studied component. So it has been identified as an effective anti-inflammatory agent to reduce the bruising and have been reported to be capable of killing cancer cells through dietary consumption or direct exposure to skin cells. So means that some people cannot adapt to the taste of the pineapple, but they need to have their nutritional punya property. They want to have their nutritional properties. So for that reason, kita boleh ambil the good things from the pineapple, we can do the product formulation. Because of the anti-inflammatory of the bromelain is very, very good, okay, for the benefit of our body, okay, to fight the cancer, to fight the inflammation, can be the antioxidant and so on. So as you can see here, this is the some of the products in the market, okay, which contain the 100%, for example, 100% pure bromelains, and also the bromelain that have been used for the wound healing. Okay, next, this is some of the, I would like to show you some of my publication about the bromelain. As you can see here, the comparative modeling studies of fruit bromelain using modular dynamic simulation. I also try to do the gene expression analysis of the bromelain from MD2 cultivars. And then I also have the review paper about the bromelain from production to commercialization. And just now I show you the analysis of the structural analysis of the bromelain from the fruit and stem. Okay. Okay, let's move to the next slide. Okay, so this is what happened. What I did in my lab is because of the properties of the bromelain, and usually for the pineapple industry, it is a huge industry in Malaysia. What happened after they harvest the pineapple, usually they will throw away all the byproducts. So from one plant of pineapple, usually only 20% is the fruit. And the other part is actually the byproduct. 20% sahaja bahagian buah. Selebihnya dia punya skin, dia punya leaf adalah byproducts. And for your information also, nanas hanya akan berbuah satu kali sahaja. So after they harvest the fruit, okay, they will throw away all the byproducts. So it will cause a lot of problem for the agriculture industry in Malaysia. So for that reason, I try to think what I can do of the bromelain from this byproduct waste. So I found that when I studied, I found that the content of the bromelain from the peel part, from the leaf part of the pineapple plant is very high. So for that reason, I try to extract this bromelain and I formulate it into the meat tenderizer product. So this is one of the product that I produce in my lab. So I call it as beeflin. So this beeflin can be used to tenderize the meat. Okay, we are doing some of the physicochemical analysis and antioxidant analysis on meat, and this is the publication. And we also join some exhibition. Okay, for example, in University Malaysia Pahang, UMPSA, and also in the national exhibition, which is inovasi dan teknologi antarabangsa. And I also go to the international level, which is at Nuremberg, Germany. Okay, International Trade Fairs Ideas. Okay. Let's move to the next slide. So other than that, what meat tenderizer product, I also try to formulate the other product, which is we call it as anti-browning enzyme, okay, for the fruit industry, where we found that this bromelain also can be used to control the browning of fresh and canned fruit. Chemicals and certain acids are commonly used. So bromelain is used as a safe enzymatic ingredient to inhibit fruit browning, for example, in fresh fruit and in dried apple slice product. So we also produce the prototype for this one, and then we publish the paper, and we also create the prototype for the commercialization. In UMPSA, our university encourage staff, okay, other than for the publication, we also encourage staff to do the commercialization. Okay, next I would like to share a little bit about my other study, which is including the medicinal mushroom. Okay, so here I would like to show you one of the study which is known as Ophiocordyceps sinensis or caterpillar fungus. So this caterpillar fungus grow in high altitude region of the Himalaya, and it is traditionally used for immunity, fatigue, stamina, and respiratory health. I don't know whether Indonesian people are aware about this type of the fungus, okay, which is kita panggil dia sebagai caterpillar fungus, okay. And it is a kind of the traditional Chinese medicine, okay. It is tonic for them. It is extremely expensive and rare, and the price is very premium actually. So what happened in UAPSA, we developed a scalable cultivation method using liquid and solid state fermentation. And we also try to do the strain preservation. What happened, it is hard to maintain the cultivation of this species because of it is survive at very harsh condition. So we try to preserve the mycelium of this species in our lab so that we can produce it, we can regulate the production. We can produce the sustainable product from this mycelium. So what happened in our lab, we enhance the bioactive compound from this species, which is cordycepin and adenosine, which is very good for the anti-inflammatory and anti-cancer potential, and they also contain the polysaccharide that can be the prebiotic and antioxidant effects. So yeah, this is the cordyceps sinensis in our lab, and this is the life cycle of cordyceps. As you can see here, this cordyceps actually survive in the host of the larvae. So selalunya dia akan bunuh the host, which is the larvae, which is we call it as the infected larvae, and they will thrive from the head of the larvae. And this is the fruiting bodies, as you can see here, and from here we try to do the cultivation in our lab, and then we for this project we work together with the local industry in Malaysia for the product formulation. Next, so this is what happened in our lab, the cultivation of cordyceps sinensis using the tissue culture technology. And this is the findings where we successfully cultivate the mycelium of this fungi in large scale, and then this the properties of the mycelium is comparable to the wild type that contain from Himalaya. Next, I try to share with you, okay, some of the other interesting project that happened in my lab, which is the tiger meat mushroom or Linosus rhinoceros. So this mushroom actually indigenous to Malaysia and Southeast Asia and traditionally used to treat cough, asthma, fever, and worms. So it is rare also because we can only find this species in the deep forest. So what happened, because of the scarcity of this sample, we try to do the artificial cultivation, okay, to faster the control biomass production. And then we do the extraction and profiling of these fungi. Okay, we analyze the polysaccharide, terpenoids, and phenolic compounds. And then we also test the anti-inflammatory, antioxidant, antibacterial properties of this enzyme. And for this project also, we collaborate with the local industry in Malaysia for the commercialization purpose. And then this is some of the challenge and opportunities, okay, to standardize the standard operating procedure, okay, to cultivate this mushroom in our lab. But it is very potential for the therapeutic and pharmaceutical industries. And this is the tiger meat mushroom cultivated in our lab. We do also the tissue culture technique. And this is the product, okay, the what we get from our cultivation, okay, in our lab. Okay, so this is actually the plot is actually at a Center of Excellence Bioaromatic, the one that Prof. Pujianto visited last time. Okay, so maybe if Prof come again, I will show you our plot. Okay, next. So I would like to share a little bit about the research to commercialization journey. As you can see here, a lot of exhibition. For example, the point to join the exhibition is actually to find the industrial partner actually for the commercialization purpose. This is the things that happen in UMPSA. So this is our product in Germany. And then this is the International Invention, Innovation and Technology Exhibition, okay, happened in Malaysia last 2 years for our immobilized enzyme for the edible bird nest cleaning. And then I would like to also show you the Malaysia Technology Expo that I joined last year, okay, 2024. Okay, I show you the mushroom products and the growth enhancer of the mushroom, which is isolated from the pineapple byproducts. And that's all from me. Terima kasih. But before that, I would like to share a little bit about my university so that if we can see the potential of the next collaboration. Okay, allow me. Okay, so I would like to, because we still have time, give me maybe 5 to 10 minutes to introduce a little bit about University of Malaysia Pahang Sultan Abdullah. Maybe this is new for our student here. So before this, our name is University of Malaysia Pahang, but after that we rebranding, okay, to University of Malaysia Pahang Sultan Abdullah in 2023, okay, which is the short form is UMPSA. And then this is our chancellor, okay, and this is our Pro-Chancellor. So this is the vision of our university, okay, a distinguished technological university. So the slogan is technology for society. So UMPSA, it is public university. We have 2 campus, one in Pekan, Kuantan, and one in Gambang. So my faculty is in Gambang. So again, it is number 1 technical university in Malaysia. Okay, 21 years. Okay, and it is top 100 to 150 university below 50 years. And this is about UMPSA, 2 campus, as I mentioned to you, and our faculty is in Gambang. Okay, but Pekan is much bigger than Gambang. So this is our focus area, and biotechnology is one of them. And I want to share a little bit about our faculty. So my faculty is Faculty of Industrial Sciences and Technology. And then this is our aim for the faculty to be center of reference for science and technology for industry. And then this is the faculty top management, me as a dean with 3 deputy dean for academic, student affairs, and also research and postgraduate studies. And this is the other head of program, or in Indonesia, I think it is coordinator, or head of study. I think it is similar. For us, we call it as head of program. And then this is the academic program. We have doctorate, master, bachelor degree, and diploma. Okay, so we have 4 programs actually, which is applied science in industrial biotechnology. We have also industrial chemistry, material technology, and occupational safety and health. Okay. And then we also have dual degree program for the biotechnology program with the Kazakh National Agrarian University, or Kaznaru. And this is about our student. For the active student, it is about 1,300 students for the active one. And for the postgraduate, we have more than up to 200 students, which is degree and master. And then we also have the publication, which is Current Science and Technology. This is our journal. And this is some of our facility, okay, in the lab. Okay, so yeah. So actually we receive a lot of inbound students. So me as a dean really encourage for the student to come to UMPSA as the inbound mobility student. We can do a lot. We have also a summit camp and so on. So at that time, we can expose you to our facilities and also to our culture. And then, before this, we also have a lot of mobility program. Okay, we send our students to Indonesia, and then we receive also the inbound student from Indonesia. So we open hand to receive Undip students and also the staff, ibu dan bapa dosen to UMPSA, especially to Faculty Science and Technology Industry. So for that slide, I will end my presentation. Thank you very much.
Okay, thank you very much, Prof. Aizi, for such an inspiring presentation. Yeah. Okay, we just listened to Professor Aizi explaining about the definition of enzyme, their mechanism, their application, and also about the protein engineering, enzyme engineering, and also including the development of keratinase from that nest. Also, it's interesting you said about the bromelain, because we use the pineapple in Indonesia to tendering the meat when, especially when Idul Adha. I think everyone is using that pineapple for tendering the meat. Okay, now we will continue with the discussion. I think there's so much question that had shown in the chat column. So the first question is from Naswa Salsabila. Naswa Salsabila, you can ask Professor Aizi directly using your microphone, please.
Hello, that's my sound. It's heard now.
Yes,
Okay,
please.
thank you.
I can hear you. Please.
Okay, thank you for the opportunity for ask the question. Hello, Professor. How are you today?
Fine, thank you. Alhamdulillah.
Alhamdulillah. Thank you, Prof. So I want to ask about. I have heard from several sources that individuals intolerant to cow's milk products have little or no lactase in their bodies. So I want to ask, from enzymatic perspective, can people intolerant to cow's milk consume lactose produce if the enzyme is broken down? Because I heard is the our ancestor is honestly didn't milk too much, didn't drink too much milk, so the lactase in their bodies is like decrease the the frequency. Thank you, Professor.
Okay. Thank you. I try to ask your question. It is about the lactose intolerance, if I'm not mistaken. Okay. So basically, lactose will be degraded by lactase. So lactose intolerance can be found in baby usually, okay, infant, okay, where some of them cannot degrade the lactose. What happen? They will vomit when they introduced by the milk. So selalunya kita akan pakai susu yang lagi mahal lah, yang lagi kurang dia punya lactose punya ataupun lactose yang sudah didegrade. So this is how industry do it. It's by using the immobilized lactose. Immobilized lactase. This immobilized lactase will be used to treat the lactose. So it will produce the milk with the degraded lactose. Okay, so the one, okay, maksudnya baby tadi yang tak boleh degrade lactose, they still boleh consume milk, they will get the benefit from milk, but they will reduce the intolerance to the lactose. So selalunya by time kita akan create enough amount of lactase, so kita boleh start consume the lactose. Tetapi based on the study, lactase, the amount of lactase will reduce again by aging. Selalu bila kita dah tua, kita akan start rasa semula kita tak boleh minum susu. Kita rasa pelik. Dulu kita boleh minum. Now bila kita minum susu, kita rasa sakit perut, kita rasa bloating. This is tanda-tanda yang kita dah ada lactose intolerance. So in the market, they have the lactose intolerance product tetapi agak costly. Oh, so this is the good things about the enzyme technology, that they will try to degrade priorly the lactose, okay, to produce the lactose intolerance product, so that these lactose intolerant people can get benefit from the milk punya product. Okay, I hope I answer your question.
Thank you, Professor. I understand.
Okay. Silakan, any other question? Saya rasa macam mengajar masa COVID.
Okay, thanks. Wow. Next, Sandra Novita, you can ask direct.
Silakan, Miss Sandra. Saya tak dengar.
To Professor I.
Saya je ke tak dengar? Macam terputus. Nurina dengar tak? Sandra?
See, Alexandra. I
Yeah.
I cannot hear your question. I'm sorry. Maybe Nurina can help me to read from the chat box.
Okay.
Okay, thank you very much.
This question from Alexandra Novita. Hello, Professor. I am Alexandra Novita, the second year biotechnology student. Considering that EBN contain complex biomolecules such as glycoprotein, IGF, and sialic acid, how can enzymatic bioprocessing be optimized to enhance the extraction of this compound while preserving their functional properties during scale-up production?
You asked me about how to enhance the properties, means that how to preserve the bioactive compound from ABN, okay, by using the enzyme technology. So for the extraction, we don't use the enzyme technology. So for the enzyme technology is basically for the treatment of the EBN. Okay, for example, that I present just now is that during the cleaning process, where we try to eliminate the unwanted compounds from ABN by using the keratinase enzyme, so that the enzyme will contain only the edible bird nest without the contamination of the bird feather and so on. So this will enhance the quality of edible bird nest. So if you want to do the extraction, you will need to use another method for the extraction purpose. And then this extraction method is also can be optimized in order to preserve the bioactive compounds, so that you will get the optimum bioactive compounds or the extracted product from the edible bird nest. Okay? I hope I
Okay,
can
is it
answer your
clear,
question.
Vanessa? I
Very
think,
active student from Undip. Okay.
okay. Okay, let's continue the next question. It's from Vanessa. Vanessa Atika. Vanessa Atika. Okay, let me read for you because the Vanessa Atika, it's not open for me. Vanessa Atika asked a question about, I want to ask some question, more than one. First question, can we use bromelain for seafood product as an antibiotic or antimicrobes? And the second question is, what is the best application of bromelain according to Prof. IZ research?
Okay, thank you, Vanessa, Atika. So based on your question, I also read in the chat box. So basically, can we use bromelain for the antibiotic or antimicrobes? For my study, because bromelain itself is enzyme, so you need to bear in mind. So it is not the metabolite, so it is the enzyme. So it maybe has the antimicrobe or antibiotic properties, but not so much. Only if for example, if the bromelain can degrade the cell wall of the certain microbe, so we can consider it as the antimicrobial properties. Okay, so that we can use it as antibiotic. Okay, so again, the bromelain itself is the enzyme. So based on my research study, what is the best application of bromelain is actually, again, bromelain is categorized as proteolytic enzyme, the enzyme that can degrade protein. So the best application of bromelain is actually to degrade the protein. So protein is found a lot in the meat, in the milk, okay, susu. So if you want to use bromelain at its best, so I mean for the commercialization, so you can use it as the to degrade the protein. Okay, so you can use it as meat tenderizer, as you mentioned. Moderator mentioned just now in Indonesia, you use a lot of pineapple, okay, during Eid Adha, okay, to tenderize the meat, right? So this is the main purpose of bromelain. But the bromelain itself also contain the anti-inflammatory properties, anti-radang. So which is very good for our body, okay, to fight the cancer, okay, can be the antioxidant, okay, for our health actually. So for that reason, there are a lot of bromelain supplement in the market, okay, because of its pharmaceutical properties. Okay, kalau pharmaceutical, that part yang you boleh guna, but kalau you nak guna untuk food and beverage, you boleh guna dia sebagai agent for meat tenderizer and also to degrade the milk. Okay, you boleh pakai dia dalam detergent product and so on to remove the protein stain. Okay, I hope I answer your question. Okay, thank you.
Okay, thank you very much. Okay, next question from Mirza. Mirza? Mirza?
Yes, yes. Okay. For the introduction, my name is Mirza, and I have a question regarding the commercial viability of this enzyme-based approach. How does the effectiveness of bromelain compare with synthetic meat tenderizer in terms of both tenderization efficiency and consumer acceptance? So I just want to know the efficiency and the acceptance from the people who consume the product of the bromelain from tenderization meat that you say before. Thank you very much.
Thank you, Mirza. Okay, to answer your question, so basically nowadays people are looking for natural products. They love to see the product come from the natural sources. So this is because they believe that natural sources it is more safer than the product that produced from the chemicals. So this is the thing. So for the consumer acceptance, I answer your question is that it is natural products. So based on efficiency of the bromelain to be used as the meat tenderizer, so as you know about the theory, we relate back to the theory. Enzyme is very specific. Okay, it will recognize the specific substrate. So for the bromelain, which is categorized as protease enzyme or proteolytic enzyme, it will only recognize protein. If we put bromelain in a mixture that contains starch, that contains lipid and contain protein, they won't disturb the lipase and also the starch. They will only found their substrate, which is protein, to do the degradation process. So if we use the product, okay, that contain the chemical product, it is very diverse. Chemicals, which is catalyst, not biocatalyst, it will degrade everything in the reaction mixture. Okay, so dia kalau dia jumpa fat pun dia degrade, kalau dia jumpa starch pun dia degrade, which is not good actually. Okay, so this is the good things about the enzyme. It is safer, it is biodegradable, it is come from the natural products, which is bromelain comes from the pineapple. So I believe that in terms of the effectiveness, enzyme is more effective than the, in term, maybe the effectiveness is comparable, but in terms of the safe usage, enzyme is more preferable. And then, this is the things that we need to consider. This is the thing that we can educate the consumers, and this is can be the product value actually. So bila kita nak jual sesuatu produk, kita kena tahu what is the value of the product. So this is the things that we can educate the people, the safeness of our product. Okay, thank you, Miza.
Okay, thank you, Professor, for the answer.
Okay, after the Mirza question, we have Salma Izzatur Rahma. Salma, where is Salma?
Yes. Okay. Hello, Professor. My name is Salma Rahma, and I have one question about the bird nest. During the production of bird nest, there is a challenging in cleaning manually, and then there is another solution which is bleach. And from the bleaching can affect or change enzymes, and is it dangerous for humans? And if it's dangerous, is there any solution?
Okay.
Thank you.
Okay, thank you, Salma. So yeah, I explained during my presentation. So usually, for the manual or traditional cleaning process of bird nest, usually there are a lot of people, okay, or it is laborious, means that there are a lot of workers that will sit together, and then they will usually, the process is that they will dip or put the edible bird nest in the water for some time. And after the bird nest is already lembut, softer, and then they will use the picker, okay, to remove the feather one by one. So pernah nampak kat picture tu kita akan pakai untuk remove the bird nest one by one. So as you can see here, the process is really time consuming. So bila ada ramai pekerja, so it is very costly actually to the industry. And then, but bila you remove the bird feather using the pitcher, so you tak akan pakai any bleacher. So if you want to export the edible bird nest outside, it is acceptable, it is safer. So usually I believe that Indonesia do the same thing. Edible bird nest ataupun Swisslet, selalunya kita akan ekspor ke China. China sangat suka makan edible bird nest because of they believe that edible bird nest is contain the bioactive that which is anti-aging. Kalau you tengok pun dekat produk dekat pasaran sekarang ni untuk produk kosmetik banyak yang ada letak edible bird nest sebagai dia punya elemen because of anti-aging properties. So kalau kat China, if you want to export the edible bird nest to the China, it need to be safer. So by doing the traditional punya process, pakai picture tu tadi, it is safer. So it is allowable to export to the China. However, some of the industries, they are using bleaching. So bleaching, for example, contain the hydrogen hydroxide, which is not good for the human and also not good to the environment. Okay, so for that reason, I come up with the technology, okay, which is the cleaning solution, which is contain the food grade components and also the enzyme that will target the keratinase. So the enzyme will degrade the keratinase, make it loose, and then the enzyme, the keratinase enzyme will target the keratin, which is the bird nest. The feather of the bird, and then this feather will be loose. Dia akan jadi pecah dan dia akan keluar daripada bird nest. So kita akan lagi senang nak cuci bird nest tu tadi. And then the enzyme, the good thing about enzyme, again, it is safer. Okay, it is biodegradable. So it is accepted for the product to be exported outside. Okay. Okay, I hope I answer your question. Thank you.
Thank you,
Thank you,
Professor.
Salma. Okay, next question is from Aima. Aima,
Yes. Thank
yeah,
you.
you can answer it.
My name is Aini, and I have a question, Professor. What strategies are being used to support the tiger milk mushroom products commercialization for wider use? Will it be a product that will be sold in local markets or supermarket chains? And if it was already sold in local market, how was the public reaction to the product? Thank you.
Okay, thank you, Aimi. Okay, so for the tiger milk mushroom, again, the native source of tiger milk mushroom only can be found in the deep forest. So for that reason, selalunya untuk wild sample kita akan dapat daripada the native people. Dekat Malaysia kita panggil orang asli. Okay, so these native people will help us to find the wild source, which is actually very hard to find. Sebab usually for the tiger milk mushroom, the sclerotium or the tuber part will be in the soil. Dia duduk dalam tanah. After 1 year, only the fruiting bodies or the cap will be come from the soil. Okay, this is the uniqueness of tiger milk mushroom. Selepas 8 bulan ke 1 tahun, baru dia punya cap, okay, dia punya cap tu, stem, batang dia dengan cap akan keluar daripada tanah. So, can you imagine, it is hard to find the wild or the natural source of tiger milk mushroom. So for that reason, but however, this tiger milk mushroom contain a lot of nutritional properties. So for that reason, in order to make sure the sustainability of the product for the commercialization purpose, then come the artificial cultivation in the lab. So we artificially cultivate the tiger milk mushroom in the lab so that we can control the production, okay, and then we can confirm the we can have for the commercialization. So for that reason, this project is funded by industry, local industry in Malaysia, that already produce the product, okay, which is the tiger milk products. So they want to have the sustainable source. So we help them to produce the sustainable source. And then after that, they will create the product, okay, which is the functional food or supplementary product that already found in the market. So for the acceptance, so because of the product is actually, the tiger milk mushroom itself, as I show you in my slide, is similar to the wild type. So the acceptance of the community or a public society is very okay, very good. Okay. And because of we as a scientist also educate them that the properties of the tiger milk mushroom is also similar to the wild type. Okay. So yeah, I hope I answer your question, Aimi. Thank you.
Thank you. That was very interesting.
Okay. Banyak
Okay,
soalan.
there's still a question from the student from Kennard. Can you please?
Okay, hello, Professor. My name is Kenneth. I
Hi.
want to ask about Glaciosima antarctica. Glaciosima antarctica has been found to contain numerous loops that play a crucial role in enzyme regulation and activity. How can this loop can contribute to enzyme regulation and activity?
Okay, okay. Thank you, Kenneth, for your question. So as I mentioned also in my presentation, I think you are what you mentioned about the loops, right? So the loops is actually the things that differentiate. I mean, the number of loops is the things that differentiate between the cold adapted enzyme and mesophilic enzyme. Mesophilic enzyme is, I'm referring to the enzyme that produced by the microorganism from the local temperature, which is the current temperature in Malaysia and Indonesia. So usually, we found that for the cool adapted enzyme, they contain a lot of loops. So this loop actually help them to be more flexible. What happened in the cold temperature and lower temperature, this temperature will reduce the movement of the enzyme. So if the enzyme cannot move, they cannot perform the catalysis. Okay, this structure is really related to the function of the enzyme. So by having a lot of loops, okay, this will help the enzyme to move. Okay, why the movement is important? Because of this movement will ensure that the substrate will, when the substrate come to the active site, the movement will make the catalysis to happen. Okay, so this is the thing. So this loop will help the flexibility of the enzyme and then will lead to the catalysis performance of the enzymes. Okay, Kenneth? I hope I answer your question.
Thank you, Prof.
Okay.
Thank you, Kennard. Also, there's a question from Kennedy. But please, Kennedy, can you open your camera, please?
Okay, hello me. Hello, Prof. My name is Kennedy Chandra, and I have a question about the caterpillar fungus. First, we know from the slide that the caterpillar fungus can be obtained by the infected insect, and that is really hard to find from the natural form and have a high price. So my question is, can caterpillar fungus directly used as an anti-inflammatory agent without undergoing extraction or special formulation? And my second question is, because this fungus is really hard to get, can we like grow this fungus by the lab? And does it really require special technique, or can it just be cultivated like ordinary plants? I think that's all about my question. Thank you.
Thank you, Kennedy. Okay, so you have two questions about the caterpillar fungus. So yeah, this caterpillar fungus contain a lot of anti-inflammatory properties, but we cannot get this secondary metabolite without extraction. So we need to use the extraction method. The simplest extraction method is the hot extraction method. Means that you put the sample in the hot water for certain time. So after that, you can get that secondary metabolites. So extraction method is required, okay, for the maximum effect of the anti-inflammatory properties. You also can eat from the crude, but maybe it is not optimum. And then after that, you ask me about, can we cultivate this caterpillar fungus or cordyceps in the lab? Yes, there is the things that we are doing now. Because of the scarcity of the sample, we cannot get, usually we need to go to the Himalayan, okay, pegunungan Himalaya in order to get the sample. So now we can, promise the sustainable source by doing the artificial cultivation in the lab. And yes, This species requires special technique and also special requirement. For example, they need to be cultivated at 23 degrees Celsius for 3 months. So it is very challenging actually for the cultivation technique of this kind of fungi compared to the other species. Okay, this is what I can share. Okay, thank you, Kennedy. Okay.
Any more question, Kennedy? Okay, I think it's enough. Okay, there are two last questions from Alicia and Sarifa. Alicia first, can you open your mic? Alisa, Dian, and Fristania. Okay, I'm gonna read the question. Hello, Professor. My name is Dian. I would like to ask, is bromelain using to the food safe for women with pregnancy? Because there are a lot of assumptions in Indonesia that bromelain in pineapple can make miscarriage.
Okay. Thank you, Alicia. I'm not medical doctor, but there is no scientific study mentioned that by eating pineapple will cause miscarriage. Okay, so I don't have any idea because yeah, maybe it will be misleading. So, but there is no scientific study mentioning that eating pineapple will cause miscarriage so far. But maybe if you consume a lot, okey, apa-apa pun kalau kita makan berlebihan akan efek kepada badan. So, maybe. Okay, because of anti-inflammation properties, maybe. Okay, thank you, Alicia.
I think it's the best answer for that
Thank you,
question.
Professor, for the answer.
Okay, the last question, last but not least, from Sharifah.
Hi,
Sharifah
Professor.
Naila.
Okay, my name is Sharifa Naila. I have a little question about your paper show us before about comparative modeling studies of fruit bromelain using molecular dynamic simulation. Does the 3D structure of bromelain remain stable, or it does show conformational change under condition or like temperature more than 20 degrees or something like that?
Okay, thank you, Sharifah. Very good question, very scientific
Okay,
question
thank you.
so far. So basically, yes, we are doing the structural prediction, and from there, we study the structure by using the molecular dynamics. Molecular dynamics is the best platform to see about the stability of the enzymes. So by using molecular dynamics, we found that the structure disrupted after the certain temperature. So molecular dynamics actually analyze the movement of the atoms in the structure. So yes, to answer your question, under certain condition, okay, which is more than its optimum temperature, the structure does disrupted or degraded, okay, and this is validated by the study of the molecular dynamics. Okay.
Okay, any further question from any participant? Okay, I think because of the time constraint, we will have to conclude today's presentation and also question. We sincerely hope this will lead to many fruitful collaboration with Prof. IZ. I think the discussion is very, very interesting, and we really truly appreciate your time and also contribution to this visiting professor online. And also thank you for all participants for your active engagement. I think we have to conclude our discussion and main session. Okay, for the last, let us give our warm appreciation to Associate Professor Dr. Aizi Normazila Ramli. Give applause, please, everyone.
Thank you very
Thank you so
much for
much.
the opportunity. I hope to see FSM students in my faculty after this.
Okay, Prof. IC, maybe we
Yeah,
can
hopefully next
do the
semester.
student exchange, yes?
Yeah, student exchange and also staff exchange.
Ah, yes, of
Wah,
course.
it's very
Yeah, I would
interesting.
like to
We
see.
will be waiting
Yeah,
for you in Indonesia also,
Bapak
yeah?
dosen in my faculty.
Oh, yes,
Hope to
yes.
discuss
Okay, okay,
further,
okay.
inshallah.
Okay, Pak Puji will agree with all this good idea, yes? I
Inshallah.
think. Inshallah,
Thank
inshallah.
you very much. Thank you
The
very
sooner
much.
the
Thank you
better,
very
yes,
much
Prof?
for the opportunity.
Yes, thank you so much.
Okay, we will return to Jordan as MC.
Okay, thank you, Bu Nurina. Now that we're close to the end of this event, but before we close this event, we have a little something for Prof. Dr. Aizi. Can we share the certificate? All right, here we have a certificate for the visit, but it doesn't have the signatures yet, but the one with the full signature will be sent to Professor in the meantime. So beforehand, can we have a little photo to memorize this event? And also for the first participant, please can you guys open your camera? All right, so okay, now that we're at the end of this event, thank you for all the participants, students, and lecturers for spending their time attending the visiting lecture by Professor Dr. Izzy until it's finished. Me, myself, as the Master of Ceremony, for this morning, I would like to apologize for any words or actions that might be wrong. Good afternoon, everyone. Wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Assalamualaikum warahmatullahi
Thank you.
wabarakatuh. Thank you so much, Prof. IC.
Thank
We
you.
will contact you soon after this, yes?
Thank you so much.
Inshallah. Thank you.
Sehat
See you
selalu ya,
soon.
Prof.
Pak.
Yeah.